It's a few minutes after 7 here, so we'll go ahead and get started. Welcome everyone. It's great to have everyone this evening, and I hope you've all enjoyed the last 3 webinars as part of this series. And they've covered the biology and some of the genetics behind what goes into these genomic enhanced estimated breeding values. And I hope tonight we can provide more details on the practical steps that we need to take to attain these genomic enhanced estimated breeding values. So tonight we have with us Tom Hodgman and Lynn Fahrmeier, both Katahdin breeders. Tom is from Maine and Lynn is from Missouri. And they've been really crucial and very involved in this whole process of developing and commercialization of this technology. And that's not to say we've had, through this whole project, there's been many hands that have been involved, and many folks, both from the university side as well as from the producer level, that have contributed and spent many hours working on this technology and getting it to where it is now. What you see in here tonight and what you've heard over the course of this webinar series is the result of the hard work of many individuals. And so I want to recognize all of those folks. And so it's an exciting time that this technology is ready to be implemented. I look forward to using it and seeing what can be done and the improvements that can be made. And so I look forward to their presentation tonight, discussing the sample collection and data submission process. And with that, I'll turn the floor over to Tom, and he's going to start us off this evening.
Thanks a lot, Andrew. I am looking forward to sharing this information with everybody. It's been something that we've been working on for a number of weeks now. Tonight's presentation is going to be a little bit different than what we've seen in the past 3 webinars. I'm going to share the floor with Lynn Fahrmeier tonight. I'm going to cover a series of slides. Should take 30 to 40 minutes to get through some slides to just go over a number of different things, and then we'll switch to a Q&A session where we can have a little bit more of an open forum and not just follow up on the chat, but we can have some in-depth conversation. So I'll try and move right along so we can have plenty of time for discussion. Tonight I want to very briefly review some of the key points that were made during the previous 3 webinars. And then I'm looking to spend most of the time covering the actual sample collection and data submission process. And I'm going to do it in a couple of different levels and step us down through where we take a little bit of an overview, and then we'll go through it in much more detail so everybody can really get their arms around it. I'd also like to close out with some follow-up things that we're working on, some things that are coming, some stuff that's we're working on right now that's going to inform the process significantly. So with that, I'm going to get underway. So in review, the previous 3 speakers did a great job of explaining a number of key issues related to genomic testing. And the first, and probably the most important in my mind, is that it'll to learn about the genomic relationships within an animal's pedigree. So you can think of it in one way that what genes a lamb inherits from its sire and its dam, but it's also could be thought of as what genes a lamb has in common with other members of its pedigree. So it's litter mates, its half sibs, its grandparents, and that sort of thing. And really, that's the power of the genomic pedigree. And I'm not going to go into any more detail in that. Dr. Lewis and Dr. Greiner did a really good job of explaining that. But I just want to bring up that point again, that we're building a genomic pedigree, a better pedigree at identifying animals with superior genetics for production traits. One of the things that's also been covered a number of times, or mentioned a number of times, is this idea of increases in accuracy with genomic enhanced estimated breeding values. And that's really driven by a couple of things. The first is it depends on our existing accuracy in our traditional EBVs. So Dr. Lewis showed some graphs that show this relationship, and he found that there was in the test data that he's run that there's a slight increase in accuracy, but it was really a function of how much the existing accuracy was. So if you've already got high accuracy in your traditional breeding values for certain traits like weaning weight or fecal egg count, you can't expect to get a whole lot more. So if you're already at 80, 85%, there's only so much more room to go. So we can't expect huge increases in accuracy. Those with lower accuracy in their traditional breeding values, we're likely to see more of a kick from genomic enhanced estimated breeding values. And another point is that it's really trait dependent. So those traits that are directly measured on an animal, like body weights and fecal egg counts, those are the ones that already tend to have higher accuracy to begin with. It's those difficult ones like the maternal side that's really where we're hoping to see an increase in accuracy. Those ones that take a long time to prove. Another point that was made by Dr. Lewis and Dr. Greiner were that you're likely to see a slight adjustment in genomic enhanced estimated breeding values over our traditional breeding values, but that adjustment could go in either direction. Okay, it's not always going to increase. We're likely to see both slight increases and slight decreases, or maybe no movement at all. But we shouldn't expect major shifts, and we certainly shouldn't expect only increases going forward. So we're building a better pedigree, and it's going to reveal some strengths and some weaknesses in our animals. The other thing that we're going to have available to us is a more robust set of genetic conditions. These are also often called causatives or causative genes. These are, many of us are already genotyping for scrapie at codon 171. I certainly do. In many cases, buyers demand it. And we're already doing that. Others that we'll be offering as part of this package are myostatin and we believe OPP susceptibility as well. There are other genetic conditions that we hope to include in the future, perhaps in the near future. So stay tuned for that. But I just wanted to cover those. That's something that is all part of this this big package. Let me just say that that genomic enhanced estimated breeding values and working towards them is definitely a multi-step process. Starts with tissue collection on farm, sample submission, genotyping at a lab, parentage and genetic conditions determination, reporting that back to the producers, then submitting our phenotypic data and a corrected genomically verified parentage on our animals. And then we'll receive reports just like we always did, but this time there'll be genomic enhanced estimated breeding values. As you may have heard, once the switch is flipped, as we say, for a breed, there'll be no more traditional estimated breeding values. But those that genotype their animals will see the greatest benefit from that. And I really want to point out an important distinction here is that these first three steps in the process are new, but the bottom two, that's what we've been doing for years. This is the old process we've done with lamb plan and sheep genetics for many years. Now we're going to tweak it a little bit. We're going to use a corrected, genomically verified parentage, not just what we wrote down in our record books, and we're going to get back these genomic enhanced estimated breeding values, not the traditional EBVs. So it's a similar process with a couple of prior steps involved is what this is. Let me walk you through what it's going to look like. It's going to start with sampling on farm, where we're going to punch a very hole in the ear of a sheep, that sample. We're going to do that several times. Take those samples, ship them off to Rusty at NSIP, and he's going to then collate them and organize them and send them to Neogen, our partner lab that's going to determine the genotype on those samples. Neogen works closely with AGBU, which is the Animal Genetics and Breeding Unit. It's part of the same entity where Sheep Genetics operates in Australia, so Meat Livestock Australia. And so AGBU will do the actual determinations of genetic conditions and genomic parentage. After that, AgBoo will return to Rusty a set of reports on parentage and genetic conditions. Rusty will subdivide that master report into reports for each individual producer. So you will receive a report for the samples that you submitted on the genomically verified parentage and genetic conditions. Then, as we've all done in the past, we've collected our on-farm data, our weaning weights, our birth weights, all those things, put that all together in Pedigree Master, together with the proper parentage, the correct parentage, and then we ship that off to Sheep Genetics, Lamb Plan. Essentially, send it all to Stephen Field, like we've been doing for many years now. Stephen Field crunches the numbers in the bi-monthly run, and then returns to the individual, directly to the producer, that those reports, the individual listing report, the sire summary, all those reports, as well as that link to download your updated database. So, but when we get those back, those will be our genomic enhanced testing values. So in a nutshell, it's 2 trips to Australia and 2 trips back to North America. And let me take and step that down a little bit for you. This tissue collection idea, this DNA sample collection, is based on this thing called a TSU, or tissue sampling unit. And that is a little vial, fluid-filled vial that has a little preservative in it. And underneath that red clip is a stainless steel circular cutter, which, when it's inserted in this blue device, it actually cuts a tiny little chunk of the ear away, pushes it into the fluid-filled vial, and then plugs it with a sealer at the end, all in one squeeze. So it's extremely simple to use and very hard to screw up. The worst thing I've ever had is one fly off. The animal jumped when I did it, and the TSU went flying. But but I could see it, so picked it up. It's very simple to use, and let me just say that there are numbers on the side of the TSU with excruciatingly small font. So for those who are who need reading glasses, you will most certainly need them when we're doing this. To aid in some of that, there's actually a QR code on the very bottom of the TSU, which can be scanned by a smartphone if you have an app to read a QR code, which may help reading those as well. I'll talk more about numbers in a moment. The source of the TSUs are a variety of vendors in the U.S., but I have to say that Allflex Gen system is the only TSU that our partner Neogen will accept, and most of the TSUs available online are the Allflex system. So it's not hard, and I would encourage you, after tonight's presentation or in the coming days, to just Google Allflex tissue sampling unit, and you're going to get all kinds of hits on that. But again, we won't be submitting there's no blood, no hair, no cards. None of that is going to be acceptable. It has to do with Neogen's machinery that is taking the tissue sample out and how it's handling it, and resealing the tissue sample back up for a long-term archive. And that's not available with some of these other things. So that is why we're using Allflex. Now, I have to make a pretty important point here, that there's a number on this each TSU and a corresponding QR code. It is extremely important, and I can't emphasize this enough, to properly match that TSU number to the animal's 16-digit NSIP ID number. If you fail to do that, there is all kinds of errors that are likely to come your way, right from parentage on. So it's extremely important that you get that right. And I would strongly suggest that you actually do a mock-up sampling session where you literally go to your handling system and have your shelf there, and you have your tools on the shelf, and you have everything ready to go, and you pretend to take the sample, and how am I going to get this number onto the, is it going to be on my laptop? Is it going to be on my smartphone? Am I scanning it into my EID reader? How am I going to do that? So really spend some time thinking this through, because mistakes at this level are going to cost you. And I'm not kidding. It's literally going to cost you money, because mistakes here will require paying to resample, retest the animal. Along with that, I would say that there are some accessories that might help you with your organization. The first is this little white box, which holds 10 TSUs. It holds them, as Lynn says, very nicely before they've been used. The little red thing fits in this little slot very nicely. And there's actually, the number is in a readable font on the cardboard right at the side of the TSU. So it's all very easy. And you could actually write it on the little cardboard box if you wanted to. So that's an option there. I will say that those cardboard boxes, I believe, are free from certain vendors. You just have to ask for them. So if you bought them in bulk, you would need to ask for a corresponding number of boxes. Second, there is a simple plastic tray, which is roughly $5 at a couple of the different vendors, and that holds 96 samples. So that's another one that some people are talking about using. I'm kind of a cheapo, and I'm thinking about maybe either a piece of blue rigid foam and drilling a few holes in it and putting a little label on the side of it to keep track of it. But I haven't figured it out totally for myself yet. But I'm definitely going to use some sort of system to keep track. So anyway, matching the number to the ID is crucial. Let me say that there's a set of on-farm sampling guidelines that were just where I just put the finishing touches on it. We've had, it's been reviewed by a couple of people right now, and I need just one last read through on it before it gets posted at nsip.org. But I would encourage you to look at this document. It's just in short, it's the on-farm guidelines for DNA sampling or something like that. And you'll see it. There's only 3 documents on there now, and there's a 4th one going to be there within the next 24 to 48 hours. That's going to cover a lot of what I'm about to say, and you may want to print that off and read it carefully over time, because there's a number of things to consider as to how to go about sample collection. The first is when, and that really depends on your management approach, your management system. Are you lambing on pasture? Are you lambing in the barn? How frequently do you have all your lambs together in one spot where you could go and tissue punch from 30 or 40 of them? What makes the most sense for your system? And honestly, marketing as well. When are you marketing your animals? And you would need this information. When are you making keep cull decisions? That's another thing. Everybody's going to be slightly different. So we want to be sure that you've given this a great deal of thought. So again, point you back to those guidelines. How many in your flock should you be sampling? That's another one that's going to take some thinking. It really has to do with the amount of data and essentially accuracy behind your animals, and we have some guidelines for that. And frankly, it's going to cost money. So, everybody's going to have a slightly different budget. And many folks have told me that they're just going to establish a dollar figure and work back. And what works. And we would encourage people to whatever way they can do it. We would try to help them find a system for that. And the most important thing, of course, is which animals to collect from. And at this point in time, we are encouraging all sires to be tissue sampled. There's only going to be a couple per flock anyway, and building that genomic pedigree and having those genotypes on file at AgBoo are key to parentage analysis. So let's just do all the sires. In terms of dams, not as rigid there. I wouldn't say anybody needs to do all their dams. That's not necessary. There's certainly a law of diminishing returns applies to sampling dams. Certainly, any lambs that you're sampling from, it would be a good idea to have their dams on file as well as the sire. But people don't need to think about sampling their entire ewe flock. I don't think that's necessary. But if we are going to sample some dams, and I would encourage you to do so, younger ewes with multiple lambings seems to make the most sense. As far as lambs, I'm going to change slides here. There's really a couple of different ways to look at it. So if you can get your head wrapped around this, it's taken me a while to do this. You sort of have to ask yourself a question, a couple of questions. Is your flock well connected genetically to other NSIP flocks? Is your flock part of the genomic reference population for your breed? Is your flock backed by multiple years of data in NSIP? If you could answer yes confidently to all three of those questions, then the recommendation is to focus your tissue sampling on litters from which you will keep replacements or sell breeding stock, okay? So those are what I would refer to as keeper litters. And in that case, you would sample all lambs in those litters, both twins, all three triplets from each of those litters. And we're doing that to really nail down that genomic relationship amongst siblings and the genomic relationships in the pedigree in general. Now, if you were to ask yourself those questions and you generally couldn't meet those, the recommendation at this point in time is to focus your sampling on as many litters as is feasible for you. And in doing so, you would only sample one lamb in each litter. And that will, at this point, reach out and get that genetic diversity for these production traits that we're interested in, and allow you to really establish that and establish those linkages over time, and then ultimately shift to the other strategy where you're really going to sample just from those keeper litters and nail the genomic relationships. I will say that when you do one lamb in each litter, I would encourage you to consider both sexes as you go through. So
Tom,
yeah,
what genomic reference population is, and do it for both Katahdins and for other breeds.
Yeah, I have to repeat that, Jim. What is the genomic reference population?
Yeah, so for those on the call, some will be part of the genomic reference population, and there are also breeds besides Katahdins on the call. So answer for both cases. Okay,
so Dr. Lewis and others mentioned this concept of the genomic reference population, and that is the foundation, the genetic foundation for this analysis. So it's roughly 2,000 to 3,000 individuals that represent the breadth of genetic diversity in the breed at that point in time, and then that forms the foundation for establishing the genetic relationships. So the linkage between the production traits, the phenotypes, and the SNPs, the genotype of those animals. So we know which SNPs are related to certain production traits over time. So, and that reference population, essentially we're adding to that by additionally sampling as we go forward into the future. But the genomic reference population is the foundation upon which this whole analysis is built. And that's been done by Dr. Lewis for the Katahdin breed, based on 5,000 lambs that were genotyped from 2017 to 2018. And other breeds are in the process of establishing their genomic reference populations right now. And I'll talk a little bit more about that at the end. But so once we have our tissue samples in hand, it's time to submit those to NSIP. And it's going to be important that there's a paper trail that follows those samples all the way from your farm all the way to Australia and back. And that paper trail starts with this spreadsheet. It's a basic Excel spreadsheet that's been created by Neogen in Australia, and we've modified it a little bit for our use. And it's pretty basic, but this idea that there's a connection between the TSU number and the NSIP number is critical, and this is where that connection is made. And there's also room for your farm ID number, as well as those that are using EID to put the EID number here as well. And the gender of the animal that the sample came from, its year of birth, and then the type of animal, because we'll be genotyping sires, dams, and their progeny. We're going to want to indicate that here. And there's a couple options for tests at this point, and for most of us, it's the genomic 50K is what we're after. Now, let me just say that, as in many spreadsheets, there are multiple sheets, and they're indicated by the tabs on the bottom. As everybody who knows Excel knows, there's little tabs at the bottom, and you can flip back and forth between your tabs. Well, this is the green tab, and the green tab is for individuals who have their tissue samples in the current batch. There are also a couple of other tabs, and those are, one is blue-ish and one is pink-ish. And the blue one is for what we call nominated sires, and the pink one for nominated dams. Now, you noticed on the previous one that you can indicate the type of animal as a sire or a dam. But on the nominated ones, these are when you have previously tested those animals. So these are for animals that are either in the reference population or at some point in the future, you've tested a bunch of your ewes, you've tested a bunch of your sires, you'll nominate them here. And those nominated animals will be linked up with the, as a possible sire or possible dam during the parentage determination at AgBoo. So this is that's about all I'm going to say on the spreadsheet, but just know that as we move forward, we're going to have some more detailed discussions as to how to fill this thing out. And it's pretty basic, but it would be a good idea that everybody knows exactly how to do that.
Hey, Tom.
Yeah.
So if those of us who have, like, say, sires that were already in Joan's study, this is where they're going to go, right?
That's correct.
Okay, got it.
Correct. Yep. In fact, these are actually used among that were reference population. So that's the way I did it and put down under notes. Yep, that's exactly right.
Okay, and like Karen and you guys who have ewes that were from that will go on the pink sheet, right? Or all your ewes that stayed in the flock that were already in Joan's study will go there. Cool.
That's correct. And any ewes that have been born since then, say 2020, or prior to that, 16, 15, or earlier, you would have to collect from, and that would go on the green sheet. So that's my understanding of the way it's set up at the moment.
So
Yep.
how do we know if any of our animals have been in that original study?
So, hi, Jan. I recognize your voice. So there is, we've talked about this, and there are, everybody who submitted data to that study has been sent a copy of animals that were successfully genotyped. So that has been done. Now, I know there's been a few issues, and Jim Morgan's been working on those. But so if you have animals that you acquired from an NSIP breeder, first off, you want to know which flocks were part of the original, the genomic reference population, or Joan's study, which fed into that. And there are 20 flocks. And so that's something that you should look into. And there's a list of that I actually have the list. It's going to make me sound nerdy, but in the Katahdin Herald, in the fall issue, 2020 issue, on page 8, there all 20 flocks are listed there, so you can see which ones those were. So if you have an animal that came out of one of those flocks and its birth year is 17, 18, or 19, and there are a few actually, I think, that came out of 2016, but certainly my flock was 17, 18, and 19. Those would be part of it. Now, there's theoretically possible that the genotyping failed, but would be a rare circumstance. So I would check check that list, and then I would contact those individual flocks and ask them to confirm that your animal was in there.
You could also contact Jim Morgan.
Oh yeah, that'd be another way.
And I could check against the master list. Also, there's a few sires that were genotyped that would not come from those 20 flocks. So you're welcome, but it's mostly going to be the 20 flocks that are, if you bought an animal from those flocks, that's where to go look.
Okay.
Do you think we could republish that list somewhere on the site or something to kind of?
Yeah, I think you're right. Yep, I think that'd be a great idea. And Jim is the keeper of the keys on some of that, so he knows more about how that was pulled together than I do. So anyhow, back to batching. Many of you have heard about this concept of batching, where we're going to 4 or 5 times each year, NSIP will submit to Neogen a big batch of TSUs for genotyping. And we're doing that to ensure that all the samples will be analyzed at one time. The way it works is Neogen is going to run samples 96 units at a time. And they don't like doing partial batches. They don't like, I don't use the word batch. So they have 96 unit trays, and they don't want to do a partial tray. It's inefficient for them to do that. So they like to have 96. And with batching, we've made an arrangement with them that if we submit as many as we can on 4 or 5 dates each year, they will run all of them, regardless of whether they fill out the tray or not. So if we weren't batching and you were to submit, say, 40 samples, say directly to Neogen, and your first 20 got included in a batch or in a run, and then the remaining 20 might have to wait until that tray fills out to 96, which could be weeks or months. And you would have some samples come back earlier than others, and it would be a bit of a nightmare. So we've negotiated with Neogen to accept these batches even if they are less than 96. And but we said we'd only do that 4 or 5 times each year. And we think that in 2021, it'll probably be closer to 2 batches, maybe 3. And these dates, the dates will be designed to accommodate producers' schedules and needs regarding marketing or making decisions on farm. That's really what it is. Sale consignment deadlines, you need to have those by. You need to make decisions on who to keep, who to sell, who to slaughter, that sort of thing. So we're looking for input on what those dates should be. We've done some homework on the Katahdin breed and learned about when most data are submitted, and that's informed some of our dates, but we'd like to know people's desires on that. Let's say too that we should all be aware that this may take a year or so to resolve these best dates. It's just not going to be. We can't have a crystal ball and figure out what's going to work for everybody. And as we add new flocks and we add new breeds, we're going to have to even be more flexible, I think, going forward. So I anticipate these shifting slightly over time. And currently, the current run for 2020, we are planning a May 1st first submission date. So keep that in your calendar, and a follow-up would be roughly middle of July. So that's the other one that we're tentatively planning on. So, but May 1st is what we're shooting for right now. So mark your calendars. Okay, costs to producers. This is something that a lot of folks have been asking about and a little bit nervous about, and we understand that. There's basically 3 checks that you need to write to get involved with this. The first is to purchase your TSUs from a vendor, and that's an independent purchase between you and whoever supplier you choose to use. And there's a number of different vendors out there, and we should also put together a list of vendors that we know of on the NSIP website. But the common ones: Valley Vet, Cattle Tags, Neogen sells, Allflex sells them, but in a bizarre way, they're more expensive from Allflex. But the TSU itself costs just over $2 a piece, and you need 1 per sample, 1 per animal, basically. So they're relatively cheap. The applicator is more expensive, $45, but it's a one-time purchase, so no biggie in the long run. So that's your first payment. Your second payment is directly to NSIP at the time that you submit the your tissue samples, and that's $30 per sample, and that covers batching, the whole batching process, and coordinating and collating and combining all the spreadsheets and all that, as well as getting reports back and splitting those back out by individual flocks, and then all the calls that Rusty's going to get for people about why this isn't the sire and I thought it was the sire, and all the sort of questions that are going to arise from this process. So that's part of it. The genotyping at Neogen is part of it, all part of the $30. The parentage and genetic conditions determination and reporting from AgBoo, that's included, as well as archiving the genotype data at AgBoo long-term is as well. So that's the big expenditure, is $30. The final one is $3.50 per lamb, but you'll recognize that one because that's the normal data fee. This is not an additional charge. This is the same old fee we've been paying. Rusty invoices us for $3.50 per lamb for data fees for running the analysis in Australia. That's what the current amount is. So that's not changed. That's the same. So at the end of the day, we're talking about the genomic enhanced estimated breeding values are roughly $32 per individual, per sample, plus the data fee. Now, if you think back to those genetic conditions, and I often think to Scrapie, codon 171, if you send a sample to GeneCheck in Colorado, it's going to be $11 to run it. And there's, costs for the blood tube and the syringe and the postage and all that. So let's say $12. That's a bit high, but let's just say $12. So just one genetic condition is $12 worth of that 32. If you added an OPP on top of that, there's another at least $11 onto that. And then you start, if somebody did parentage as well, like through flock 54, that's a significant chunk. So really, if you start adding up all these additional genetic conditions, they make up a significant portion of that value. And you're getting genomic enhanced estimated breeding values on top of it, which is really why we're doing this in the first place. So I just want to point that out, that when you see that number, think back to you're already paying, many of you, not everyone, but many of you are already paying a portion of that in genotyping costs for genetic conditions or parentage already. So enough on costs. So there will be established a genomic certification program, and it's going to be pretty simple. Only sheep that have actually been genotyped can be certified, NSIP genomic certified. And we have it on good authority from our Australian partners that the individual listing report and others will indicate that that animal has been genotyped. And we've investigated from our contractor for the searchable database, and we believe it's going to be fairly simple to have an indicator on the searchable database as well that shows that the animal was successfully genotyped. We're already getting, in our data that we get back from Australia right now, there's a field on there, whether or not it's genomic information, and it's essentially says no at the current time, but as each breed establishes their reference population and we, as they say, flip the switch and go from traditional breeding values to genomic enhanced breeding values, we will be able to receive that information that comes into Rusty and then goes to create the searchable database. And there we have this nifty promotional logo, which you can use to try and impress your buyers and other things at sales and for your marketing, and recoup some of that $32 per sample that you paid to genotype them in the first place. So some of the next steps that are involved here as I begin to wrap up, I'll say that there's a test run of a batch of roughly 96 samples right now in the system. We decided to do that because we wanted to see if there were any sort of glitches in the system and what needed to learn from, any problems that we found. At this point, those samples, I heard as of 3 o'clock this afternoon, those samples were had been successfully genotyped at Neogen and were electronically sent to AGBU this afternoon. So now we're waiting to hear back from AGBU on genetic conditions and genomic parentage. So we're really pretty excited to get that back and looking forward to see what issues crop up. There's already been a couple that it's revealed, and we're pretty thankful for that. As many of you know, the Katahdin breed will be the first up for genomic enhanced estimated breeding values as a result of our of the work of Joan Burke and others that have and Dr. Lewis to create that genomic reference population for that breed. And as a result of that, we're going to start. Many people are getting anxious in tissue samples and submitting, and we're planning another webinar, not really a webinar, but more of an open discussion of the sampling and submission steps. So, you know, the strategies for sampling and then the spreadsheet submission form, and we're going to do that on April 30th. Andrew kindly circulated an announcement and a link on that today. So please sign up for that if you're a Katahdin producer and you're considering submitting samples.
Real quick, Tom, the meeting next week will be March 30th.
I'm sorry, March 30th. Yeah, hey, that's a good one, huh? April 30th. Sorry, thank you. We're also thinking about a second producer meeting. Probably that one would be in April, or maybe a little later, and that would be covering any issues that arise from the first parentage reports as we get those back, because we anticipate there being a lot of questions about that, trying to be as transparent as possible there. And for folks that are in some of the other breeds, Suffolk, Polypay, and Rambouillet, those are the ones that I've heard are talking about getting involved with genomic enhanced estimated breeding values and begin building their reference populations. I know the Rambouillets will be submitting some samples this summer, at least that's what I've heard. Suffolks and Polypays perhaps as well. If you have sheep of any of those 3 breeds and you'd like to learn more about possibly submitting sooner rather than later, I'd recommend you contact your NSIP breed representative, get details on sampling.
Tom, I would add
So,
I'd add Targus to that list.
Yep,
I think Targus will be moving forward as well as those others.
Okay, great. Thank you, Jim. So to close out here, I wanted to just say thank you to a lot of folks that have helped out with this whole project. And certainly all of our speakers and Andrew for coordinating. You know, Dr. Lewis has really been a game changer when it comes to this. You know, big thanks to Joan and Jim for all the work on the Katahdin, getting the Katahdin, those 5,000 samples together. That's huge. You know, we have these commercial partners internationally on this project, so that's been key to work with them. And then the different organizations and committees that have really put in time. So as Andrew said at the beginning, there's just been a ton of effort this winter going into this, and really thankful for everybody working together on it. It's been very cool. And let me just leave this slide up when I'm done. But so here's the link to the NSIP site. And at the moment, this nsip.org under resources, genomic enhanced EBVs, there are links to all there are links to all 3 webinars so far, and I'm assuming there will be a link to this one as well soon after this session ends. And then there's 3 documents up there right now. This overview of genomic enhanced EBVs, sort of give you an idea of, if you missed it, sort of the background and justification for it, some basics on how it works. And then a report from the NSIP Technical Committee, the NSIP Genotyping Guidelines, that's there as well. And then a document that I refer to as the framework, number 3 here, that's essentially a written version of the presentation tonight, which goes into much more detail into a number of different topics. So that's something that you might want to print off and have in your office, or just know that it's there for future reference. And then, as I mentioned before, there's a document on on-farm DNA sampling strategies, and that will be posted in the next, I would say within the next 48 hours, to review that and give it some serious thought. I would recommend a cup of coffee on a Saturday or Sunday morning, and looking through that and giving it some careful thought as you go forward. So at this point, I'm going to I'll leave this up here, but we'll shift gears and put Lynn on the spot, and he can run the Q&A session, and I'll keep an eye on things from there. So.
Okay, well, wow, Tom,
Thank you.
Thank you very much. That's you covered a lot of material, and I can update everyone that on this list that's on the NSIP website, you can also download the submission spreadsheet now. Rusty has posted that during the webinar. So the biggest, I think, question maybe that came in that we need Rusty to clarify. I hope Rusty can unmute himself. We've got a question about what happens if you're, sampling someone that's a lamb that's going to be listed as a progeny, but you've already used it as a sire. And so how many different forms is that going to take, Rusty, if you can explain that?
Yeah, so if it's a new animal that's being submitted for genotyping for the first time, it will be listed on the green sheet that Tom had mentioned earlier. So it's being submitted for the first time. You can, in the column where you indicate whether it's a sire, a dam, or a progeny, you can indicate 2 different things on that. So you can put in a progeny and sire listed. But the second form, actually, it will not go on the nominated sire list, because that's only to be used for animals that have previously been genotyped. So that animal should still just have one entry on the green sheet.
So Rusty, if we're coming up to a breeding season, we should sample a sire in the fall so that it'll make it easier in the spring when we have lambs.
Yes, and I think the recommendation that I know Dr. Lewis has made it, I think some of the other presenters have as well, is to take TSUs or sample your rams prior to putting them in with the ewes, just because you never know which ones are actually going to make it home. So I would recommend taking it, prior to actually when you actually need those results.
And while we're on here, and this may be a Ron Lewis question, you know, and the fact that Jim Morgan here, has seen more of the results, do we expect more or less DNA failure with the TSUs than we did with the blood cards?
Sorry, I'm being beaten by the technology. I finally figured out how to unmute myself. Jim, I think the TSUs are actually going to produce a higher quality DNA sample. So my guess is in terms of that part of DNA extraction, it will be better. So if anything, my bet will be a higher quality starting point and therefore a higher quality genotype. I don't think we're going to lose anything.
And that would have been my scientific estimated guess as well. And while we're at this as well, Tom gave, this was actually for Rusty, Tom gave a date of 5/1 and 7 in July for submission of DNA this year. So is that the date that he's sending the Neogen, or is that the date that it needs to be in his hand? And so that's a Rusty question.
I don't know if we have discussed that. I would say that the sample definitely needs to be in my hands by that date.
But
when, Tom, was that the thought? Is that what you guys are thinking?
That's what I was thinking, but I don't know.
so it's a date for Rusty, not the date that he needs to submit the Neogen.
It's the date that, my understanding was it was the date that everyone, all the producers need to know that they have to be in by. And then Rusty has a couple of days to get it organized, to get it over to Neogen.
Okay.
That was my understanding, but I don't think we've had a detailed discussion of it, but.
I think, you know.
Okay. The reality is it's going to be like one day, one way or the other. Because, I mean, if your deadline to get them to Rusty is the 1st, I'm expecting him to mail them on the 2nd. And maybe I'm just really hard about this, but we don't have time to dilly-dally around with this, guys. We need to keep pushing. So don't wait until the last minute like you normally submit emails to Stephen Fields.
I was mostly coming at, if I get them to Rusty on the 1st, that's not soon enough. We need to get them a few days before then.
Yeah, I think we should assume that to begin with, Jim. Thank you.
Yeah.
I mean, even with shipping delays, I mean, just mark 2 weeks ahead of that date on your calendar and send them April 15th at the latest, and you know they'll be there in time.
And with the TSUs, you should be able to submit by U.S. postal mail and not have to use any overnight or FedEx or UPS. So I think that's a savings for the producer.
So they actually get delivered.
There's a question in the chat about goats, and you know, this technology works for goats as well. And the issue with any breed, number one, it has to be done by breed, and then the main issue is coming up with this reference population of 2,000 to 3,000 animals, and within a short period of time, and maybe like 3 or 4 years. And so, you know, is there funding available to do that? So that is where, you know, we want to work with all breeds of sheep. We want to work with all breeds of goats. Currently, we have trouble getting goats to even submit their phenotypic data, and we're working on trying to improve that. So, but unfortunately, this technology is breed specific. So, okay, any other questions? I can ramble just a little bit. I just want to go back and touch on what Tom said and maybe just highlight a couple things. And
can I ask a question?
Yeah, go ahead, Roxanne.
I'm having a hard time typing out what I'm trying to say. On the ewes from last year that we kept that were not sampled because they weren't in that original 3-year gap thing, and they lamb this year, should we send in samples of those ewes ahead of their progeny? Because the progeny would they be able to connect the progeny with the parent?
Rusty and Ron can correct me if I'm wrong, but my understanding is they can connect all at one time. If they're on that form, the form is, the data is put into the database, and then they do the searching to see who is the parents.
Okay. But you wouldn't want to do the progeny without doing the dam.
Well,
Sires for, yeah, go ahead, Tom.
and Roxanne, you would still get sire even if you didn't.
Yeah.
And so it's worth submitting just for the sire, and then if it doesn't make sense, if you have a dam that has mismothered a lamb who was bred to a different sire than the discovery sire, that would help you say that, figure out whether that lamb actually belongs there.
Okay.
And it's best if you have both sire and dam, and that's where Tom's guidance of doing young dams in 4 to 5 years will have your whole flock. I mean, that's the goal, is that in 3 to 5 years, you'll have your whole flock.
Yeah, that's exactly right, Jim. And I would say that, at least my understanding is that that we should put an emphasis on, at least in the Katahdin breed here, should put an emphasis on these ewe lambs we kept from 2020 that are going to start producing, either they're already producing or they're going to produce next year. Those would form a bridge to the current 2020 crop, back to the reference population. And my understanding is that's a pretty valuable connection to have. And we'd hope that everybody would sample those.
The only value in your 4 to 5 year old ewes would be if you have mismothering. A 4 to 5 year old ewe should have minimal increase in accuracy.
So I did have one quick question on the submission form for our nominated sires, nominated dams. Do we need, if they've already had a sample submitted, it's already in the database, it lists the sample ID, the TSU barcode, as well as the NSIP ID. Do we need to fill out all those pieces of information, or once that sample is in the database, can we just list the NSIP ID for that sire and have that be the link? I'm just wondering if I need to, if I use a ram from Tom, for example, do I need to sort out and figure out from him what that TSU barcode was back when he submitted that original sire sample? Or can I just list the sire ID, which I would know from a sale catalog or the searchable database? And
I'm hoping Rusty unmutes himself and answers that question. My understanding is the NSIP ID is enough. And okay,
sorry,
I guess
you repeat that? I had a screaming 2-year-old that I wasn't able to get.
The question, Rusty, is on the Andrew, you restate your question, please.
Okay, so on the nominated sires, nominated dams form, it has the TSU barcode listed as a column and also has the NSIP ID of that animal listed as another column. And I was just curious, once that animal has a sample in the database, do we need to include both the barcode ID as well as the NSIP ID, or will that automatically be associated with one another once that sample has initially been submitted? so that I could, if I used a RAM from someone else, I could just put the NSIP ID for that individual rather than having to go back to them and figure out what that original barcode was.
Yes, if an animal has already been genotyped, if it's already in the system in Australia, then all you would need to put in is the 16-digit NSIP ID number. You don't have to put in the TSU ID number into that column. And I would advise you, unless you are 100% certain that you have the old TSU number 100% correct, I would advise you just leave that column off of it. In the ideal world, we'd have two ways to look up that animal in the database that's already there, but the only one that we need is the 16-digit NSIP ID number.
Okay, perfect. Thank you, Rusty.
Thanks. And just to expand on that just a little bit, so everyone understands why we have to have this nominated sire and nominated dam list is the computer in Australia does not search the entire database of all animal genotypes to determine the sire or the dam of a lamb. It's just going to see if in your list, if there's any matches to the progeny on your list. And so that's the reason why you need to nominate previously sampled animals so they know which ones to look at to see if it's a possible match.
So this is Jim Morgan. In looking at my flock, in 2019, I have a ewe who's predicted as the dam of 5 lambs over 8 weeks. So, this is just as a warning that, as we go forward, the more animals you have in the database, the less that will happen, because right now she's related to a lot of animals, and but she's not the dam of all those. So we need to keep submitting animals that we're using, keep submitting dams that we're using, and make sure it makes sense. I think that's and trying to help the producers that were part of the Burke-Lewis grant. I know it's important to note that there are potential problems, and we have 3 rams that were submitted as the sire of 30 to 60 lambs that are not the sire of 30 to 60 lambs, or at least the sample that's submitted. So, and we need to keep that verification, the accuracy that Tom has talked about is incredibly important. And just, it'll become obvious as each of us submits, that we have issues. And I was recently working with a flock that had FarmWorks who scanned the 20 to 30 ewes that went in with a ram, and then we came back with genomics that said, no, the rams you submitted in pedigree master didn't match. And she was able to go back to FarmWorks and find that, yeah, you're right, that. So I think, genomics is going to be great. It will help correct, and I know from my own flock that a ewe that I've now eaten rejected a lamb that wasn't hers. So, she was rejecting a lamb that I thought could only be hers, and that sort of thing. So genomics will help.
Cool.
And there's all sorts of things we need to work on.
Yeah, thanks, Jim. You basically said what I was going to say, and that is I mean, in my own personal experience, last year I used Flock 54 for parentage on a large percentage of my lambs. And I was running 12 to 15% parentage errors. And I mean, I think I'm pretty good at keeping records, but some of it was cross fostering in the barn that happened before I got to the barn. And some of it was at breeding time. Kind of like Jim's example, I have to accept total blame that there was 3 ewes that obviously ended up in the breeding pen with the ram I did not intend them to go to. There's no way that I can come up with any other explanation than I just opened the wrong gate when they were running down the chute. So I've said this before, we have to give everybody a little grace. Mistakes happen, and sometimes we have to give ourselves some grace and just admit that we made a mistake and be glad that we now have the technology that we can catch some of these.
So 3 to 5 years in the future, when we have more dams in the system, you know, there's several of these there, you know, when the genomics says this is the closest relative to be the dam or the sire, as we go forward, those will be a lot more accurate.
They will be more accurate. And I think not only can we help ourselves by being more diligent as we sort into single sire mating groups, but like Tom said, it's critical that the right TSU is matched up to the right animal. And however you do that, whether it's a spreadsheet in the barn, on a computer, or on a piece of paper, or however, you're transferring data back to that submission spreadsheet, I think there will be a lot of errors that occur there if we're rushing through the process. So,
and while most of the breeders on this webinar are Katahdin breeders, you know, with my 10 to 12 years as NSIP president, I really do think that we can add Taris, Polypays, Rambouillets, and Suffolks to genomically enhanced EBVs in 2 to 3 years.
Yeah, I agree, Jim. And that's the goal. It takes funding, and there's quite a few grant applications being floated around to help those breeds. And that's where Tom was so accurate in saying, you know, talk to your breed, your NSIP breed coordinator, because they're the ones that know the most about each individual breed and the opportunities and potential of moving forward. So,
and for those Katahdin breeders that weren't part of Joan's grant, please contact Joan and I. And we will work with you to help you move forward, receiving the most impact from your genomic enhanced EBVs.
well, thanks to all the researchers that worked on that Katahdin grant, because all Katahdin NSIP members now have a new tool at their disposal because of the work of those researchers.
So it can be show clock, it can be whatever. I know you can get value on genomic sampling if you're, and you have to submit NSIP data, phenotype data, but you can all receive value, and as a past NSIP president, I'm looking forward to adding Polypays, Rambouillets, Suffolks, and Targies, because those breeds have the data to go with this. So, and if you look at Peep Extended Industry, you know, Andrew Weaver, who's helping with this, and we all need, whether it's Western, Eastern, or whatever, to all pull together to help with the sheep industry. That's all for me. I've talked enough.
It's all very true. Thank you, Jim. Are there any other questions from the participants? We still have 32 of y'all online. I appreciate that. That's obviously there's a lot of interest, and we don't want to gloss over anything here. We one of the frustrations that I think Tom and I have had, you know, we're just trying to shoot, you know, dovetail into a system that's already been put together in Australia. And back in September, when we started down this path of trying to commercialize the research that the researchers have done such a good job on, I never dreamed it would take this long or this many hours. But the reality is the production systems in Australia are different than production systems here. I hope you all see that we're not telling you that there's a standard template that you have to follow. Each farm here is different. And so, you know, we're putting out guidelines and things for you to think about as you develop your own sampling strategies. And, you know, it's kind of a spectrum. And so you, within your context of how much time, how often do you touch your lambs, how much money do you have to spend? Those are all things that will adjust how many samples you take and when you take them. And so we're trying to be as flexible as possible.
Hi, this is Jan Turner again. I've got a question for Rusty. On submitting the samples, if I've got more than 96 samples I want to send in, would it be helpful for you if I put them in that blue box that Tom was showing earlier and send it to you in batches of 96?
I think that's a logistical thing we'll still need to work out. I think you can send in as many samples as you have, which is, you know, the point of us doing the batching. So I wouldn't, if you have more than 96, I wouldn't limit yourself to just sending in the first 96 just for simplicity's sake. The more organized they are, the easier it will be on my end. But like I said, if you've got animals you want to get genotyped, regardless of how many they are, I would send them all in at one time.
Okay, thank you.
And it might be good to just touch on TSU handling and storage. So if you have a group of lambs, you know, maybe they're born in 1st of January and you want to sample them, but the batching deadline's not till May 1st.
Yeah. Yeah, thank you, Andrew. That's great. These TSUs are very robust. I've used them now for 2 years, and they cannot be in sunlight. That's the main thing. So once you've collected the sample, don't just lay them. If you're working outside, just don't throw them on a tray out in the blazing sun. Keep them in the shade. Once you get back to the house, room temperature is fine. They do not have to be refrigerated. They do not have to be frozen. So yeah, they're fairly robust little units. I mean, I think I don't know how many hundred I've used. I had one that did not seal properly and the liquid came out of the tube. And that's really the only failure, I think, of a tube that I've had. So the system works really well. But yeah, I'd say the main thing is don't leave them in the sun. Just keep them at room temperature until you're ready to ship them to Rusty. So, you know, you don't even need ice packs is the great thing about these is that, you know, you can ship them to Rusty. And I guess maybe if we're shipping in February and it's 20 below, maybe it should be an insulated box, but that'd be about my only concern.
Okay, thanks for that. Are there any other questions?
So Lynn, you think cold temperatures are worse than 70-degree?
I don't think we know what freezing and thaw cycles do, Jim. I don't know that cold is bad, but I don't know if we repeatedly freeze and thaw, what will happen.
So that'd be good to learn, as we go forward.
There's the cattle breeds are, I mean, I can just tell you that after the samples, after the DNA has, the first DNA extraction takes place, they're freezing a lot of their samples for long-term storage, partially because we don't have long-term studies yet. This technology is within the last 5 years. And so I think we'll be finding out here in the next few years, some of those answers. But for right now, just to get them to Rusty and then have him get them to Neogen, it's a pretty robust, just keep them at room temperature and out of the sun.
Are there any other questions before we wrap up this evening?
Hey, this is John Bear. I have a quick question. I've read and I was trying to find the actual article on it, but as far as sampling newborns, is that acceptable because of the potential for cross contamination? I've heard some people mention that, and I know I've read some cattle articles where cross contamination, at least theoretically, could be a problem, and I know it was a concern with some of these as far as with the Scrapie program and things like that. So any thoughts on that, whether it's okay to sample while they're, wet lambs? I had read that could be an issue. That was a question I had.
So John, this is Jim Morgan. So how have the cross contaminations?
Are you just thinking like maternal DNA from the placenta kind of on the lamb or something if they're wet newborn?
Right. Yeah.
Yeah, that's a concern. I want to know the answer too, because that's exactly what I was planning on doing, is sampling them right at birth when I tag them.
The placenta, though, isn't maternal.
Well, it's a kind of a shared organ,
Huh?
and you can have. It's a little loosey-goosey in there, but that's a question that I had, and I'd read an article and I was trying to find it here on my phone before I asked it, but they said, that was an issue, but I was wondering if anybody had heard or asked, if they'd said anything from Allflex or GeneSeq, either one.
Yeah, I mean, based on the Neogen document on the do's and do nots of TSU utilization, it suggests, not to sample. This would be in reference to cattle, but do not sample wet calves because the placenta and fluid could cause a failed sample. So I guess get them dried off as best possible and minimize, any chance of that contamination. So maybe you can wait 12 hours or even wait 4 hours until that animal is relatively dried off and cleaned off.
Or should we maybe swab them with disinfectant or something, that ear section, before we do it, if we are going to do a pretty young lamb?
This suggests do not use anything to clean off the animal or clean off the site before taking that sample.
Okay, food for thought.
I'll post a link to this PDF in the chat.
Yeah, that's the one. That's probably the one that I had read, and I'd seen some emails where folks were considering that strategy, and that was.
Yeah, thanks for the call out, John.
Yeah, thanks, John.
Add that to the list of things we have to consider as we go forward with this. One more.
So 12 to 24 hours should be pretty good.
I think once they're dried off.
Okay, so if you're all looking at the chat, Andrew posted the link for that document. And then John Coward asked a question. What happens if we find out, basically, I'm going to paraphrase this, John, correct me if I'm wrong, but if you buy a RAM from somebody and then subsequently you find out because of parentage testing that the pedigree is wrong, what happens? And that is something that the KHSI board of directors are going to have to tackle.
And I got another question in the chat. It was a direct message, but I think it's relevant for everyone. You know, when we find a parentage error, how are those errors handled? At what level are those handled? And those are handled by the individual producer. That data is going to come back to you. You need to make those corrections in Pedigree Master and resubmit that with the correct parentage so that that can be, you know, correctly applied. And so that will be handled, you know, at your level, at the producer level, not at NSIP level.
Yep. Yeah. That's a great point. Oh, and Andrew, Michelle says, if you're seeing that, Michelle sent you a message.
Let's see here.
Okay, there's two things that I want to address there with what Andrew said about correcting the parentage. Number one, NSIP cannot correct the parentage because we don't have access to your raw data. We're pretty proud of the fact that we do not have access to your private data. And so it's important for you to correct the parentage in Pedigree Master yourself. And then number two, we've been, as I understand this from the scientists, if you do not correct the parentage, then there will be a blending of what the genomic parentage is and what you're reporting. So your EBV will not be as correct as it could be if you would go ahead and correct the parentage.
Jim Morgan here. It's worth pointing out that some of the suggested parentage don't make any sense. They're basing it on the closest DNA they have, and so we do need to look at what they suggest as parentage and determine if they're right. And it includes both what some potential errors at Neogen where they put the wrong sample in the wrong, which is going to be incredibly rare, but a possibility, and we do need to point that out. But I think there's going to be chances in what they're suggesting a dam or sire that's not possible. So we do need to keep that in as a possibility. I'm done.
You're Lynn.
Yeah. Okay. Yeah. So much. Thanks, Tom. To Jim's point, that's why we're running 96 samples through the system so we can correct and we can figure out some of these quirks that come back and we can provide advice. And then just to close out my portion tonight, Andrew, thank you for organizing these. And I just want to thank Tom for putting the presentation together, and thanks to everyone that stayed around this evening.
Well,
thanks for everybody calling in. Yep.
And thank you, Tom and Lynn, for all your work on this and presenting this evening. For those Katahdin producers that are interested in submitting samples in this season and in 2021, you should have received a link to a webinar, a meeting next week that Tom mentioned for further discussion on that. So keep that in mind, and we hope to see those folks next week. For the rest of you, feel free to reach out with any questions that you might have as we move forward. Thank you for joining us over the last month or so as we've covered these webinars, and all those recordings will be posted and can be found on the NSIP website under the Genomic Enhanced EBV page. And so with that, thank you, everyone. Thank you, Tom and Lynn, and everyone else who's made this possible, all the other speakers over the course of this series. And I've enjoyed it and learned a lot, and I hope the rest of you have as well. So with that, have a good evening, and good luck with the rest of your lambing season and your spring.